I was here at the WIN Symposium to discuss our work on neo-antigens, mutant epitopes that occur in human cancers and that can be recognized by the immune system. And I think what we're seeing is that in patients that respond to immunotherapy, that the T-cell-based immune system can generally see these variant epitopes on cancer cells as being foreign and react to them. The main basis to be interested in this work is that we now know that cancer immunotherapies can be highly active within the clinic. We do not really know what these T cells that mediate these effects need to see on the cancer cells in order to be able to kill them. And in our work, we're testing the hypothesis that what these T cells see is mutant epitopes, variant epitopes, that arise as a consequence of the DNA mutations within the cancer cells. So most of our research over the past few years, we have focused on melanoma patients treated with different types of immunotherapy, either cellular therapy or checkpoint blockade. And in those patients, we've analyzed how frequently the immune system recognizes these mutant epitopes on the cancer cells. And what we see is that in most of the patients, there is an immune response against these mutant peptides that occur within the cancer. So I think with our current work and work from Steve Rosenberg at NIH, it is now quite clear that recognition of these mutant epitopes is a very important ingredient of cancer immunotherapy. And I think that really sets us up for the next stage, where we try to specifically enhance immune reactivity against these variant epitopes. So there will really be a form of personalized cancer immunotherapy. So most of this research has been done in melanoma because traditionally we had the biggest hope that immunotherapy would work in melanoma. We actually now also know that immunotherapy can be active in tumors such as bladder cancer, lung cancer, head and neck cancers. And actually also in some of those tumor types we do see evidence that the immune system reacts to this kind of mutant epitopes. So we're now slowly moving away from melanoma towards these other tumor types. Yeah, so I think there's a strong rationale to do that. I think one rationale, actually looking at our work, would be that these additional therapies could sort of serve as a way to alert the immune system towards on the cancer cells by liberating antigen. So synergy with checkpoint blockade is something that may potentially occur, and I think that deserves to be tested. In addition, I think it's going to be really important to test whether we can develop more targeted strategies to really induce immune reactivity against mutant epitopes on cancer cells. I think at this point in time, it is quite clear that in immunotherapy, T-cell reactivity is of significant importance. I think it would be quite nice to also test whether in targeted therapies or classical chemotherapy, actually the immune system also plays a significant role. And I think on the basis of the work that's emerged over the past few years, I think this is likely to be the case in at least some patients. I think the final take-home message from our work is that the immune system has evolved to fight an enemy that evolves through time. Pathogens. I think one of the issues in cancer therapy has been that tumors also have a tendency to adapt. I think the immune system actually is a very nice way to deal with that adaptation.